All right, but she asked me to talk a little bit about what can be the useful applications of these DNA taxonomy, for example, where we sequence things to understand what is out there in the world. And currently, if we use DNA work, molecular biology for taxonomy, you might get directly the idea that we are talking about so-called DNA barcoding. Have you heard about DNA barcoding before? Yes, okay, great. Who has not heard about DNA barcoding? Who has heard about DNA barcoding? Who did not yet raise the hand, although this was a yes or no answer? Okay, so anyway. So DNA barcoding is good that you've heard already quite a lot about that, is certainly, to recapitulate this a bit, to understand the huge diversity we have out there, just without being an eminent taxonomist. You know, out there, there's something like 7-8 million species. Only 1.5 million species are yet described. So you see, there's still a lot to do for us and to understand out there. DNA barcoding tries to make this a little bit easier. And DNA barcoding just aims that even a layman, so somebody who's not an expert, at least gets an idea what is out there and what he or she just collected, and to contribute to understanding of the fauna. A method introduced by Paul Hébert, a Canadian, in 2003, who was an almost retired ornithologist, who suddenly noticed, hey, I have this fantastic marker, I have this fantastic DNA region, the cytochrome oxidase 1, I use it for phylogeny, and I see this is also specific for species. So why not taking this as one of these supermarket barcodes, yeah, instead of these black and white bars, just taking these bars in A, C, Gs, and Ts and using this to understand what's out there as species. So based on this, the idea originated, we can now take all kinds of collections and barcode them, take a unified marker for sequencing, and get a database out of this, which we can use if you go out in the field and collect something which is unknown to us, to compare against these against these already known references. So far, so easy. Therefore, these DNA barcoding try to aim to find one single marker, a genetic marker useful for basically everything. Didn't work that nicely for everything. However, for some organisms, it works nicely to just use one universal marker. The idea of this marker was actually that it should be present in all organisms. Yeah, so we cannot take any kind of fancy protein that the bacteria don't, but we do. Must be easy, so ideally lots of copies. Yeah, must be also ideally useful for phylogenetic analysis. Yeah, great. Ideally, hardly or no variation inside a species. The huge variation between species. So we know, oh, this particular barcode, this particular genetic diversity just means this is just species A, and species B is already a couple of % genetic diversity different from here. So this worked then. So Paul Hébert tried that with his butterflies first, and he noticed this works great with all kinds of arthropods, other arthropods, or insects, and then he extended it to other arthropods, and he tried this with vertebrates, with fish, mammals, whatever. And so for animals, this region, the cytochrome oxidase 1, or just a 6-month base pair fragment of it, got the barcoding marker for at least bilaterian animals. Plants use something else, chloroplast genes, bacteria take something else again, 16S ribosomal RNA. Usually, you know in advance if you deal with a bacterium or with an animal. So therefore, switching here between markers is not so much of a problem. So far, so good. The idea is then already 20 years ago that we can go out, like in science fiction, with a fancy with some fancy equipment, yeah, like maybe some of you might know him from enterprise. He had this kind of tricorder. He could go out then, use this tricorder and say, hey, this is a new species, or this is something else, just taking this in the field. Ideally, having something like a handheld barcoder, where you just put a little bit of tissue into an opening, and directly all this molecular biology, trained magic, occurs, and you'll get directly the idea what it is. Maybe even as an iPhone app, yeah, of course you have to pay for it, $1.99. You just put your sample in the microphone opening, yeah, and with a little luck, and after payment, of course, you'll get your barcode, and people directly know what it is. Unfortunately, still fiction. I know that the companies are busy to have the sequencing stuff as small as possible to bring them out in the field. The next time you're somewhere in the jungle or you're at the beach, so that you could take a sequencing equipment with you, handheld, it still takes a while. Until then, we still have to do it the classical way, as taught, extract in the lab, and then PCR amplify in the lab, sequence either in the lab or somewhere else, and then just putting this into the database. But then, when you have the barcode, you know what we have there, ideally. If you have a bunch of beetles, you don't have to prepare all characteristic organs. Mostly with insects, the genitals are taxon specific, yeah, and you would have to prepare them out of this organism, so you destroy the organism. And then you need to be expert to say whether this is different from this, or from this, from this, from this. Then you just take a little drop of hemolymph, or whatever else of tissue and cells you get from the beetle, and compare them with each other, directly then seeing that these 5 guys, although they look the same, are pretty much different, probably different species. Or if you find some organisms you cannot associate what they are, like these guys here, yeah, cnidarians. The polyp life stage has been known of this particular species. The medusa life stage has not been known. However, from another, the medusa life stage has been known, and the polyp of it hasn't been identified yet. Yeah, you know, cnidarians have these 2 lifestyles. Yeah, one sessile polyp, yeah, asexual. Medusa are then the sexual lifestyle, which is then somewhere then also drifting in the ocean and therefore distributing the species somewhere else. Now, barcoding can tell us that actually these 2 guys, which have been described as complete 2 different things, are pretty much identical. One of the most famous are these kind of butterflies, which in the UK were pretty characteristic and have been monitored for quite a while. The skipper butterfly and barcoding, although they pretty much look the same, directly identified that these are 10 completely different species. And of course, every species by itself has its own ecological niche with its own ecological requirements, and maybe each of them needs its individual way of protection. So therefore, in this respect, DNA barcoding brought us much more insight. So far so good. Based on these knowledge, lots of different barcoding initiatives have been formed under the umbrella of the Barcoding of Life project, including, of course, sponge barcoding project. I work on sponges, we are running a sponge barcoding project, but other things, birds barcoding initiative, or insect barcoding, fish boil, or regional barcoding initiatives like the polar barcoding, where it doesn't matter what you look at, as long as it's coming from the Arctic or other polar regions. This is going to be barcoded in order to understand the biodiversity there and the links between all these kind of organisms. So far, so good, so easy. So within a couple of years, ideally it's been thought that we can barcode everything. Why did we start with the barcoding? End of the 1990s, the human genome was sequenced with a huge effort. 1999, 2001, they have been published. And afterwards, there have been these huge amounts of sequencing machines, somewhere in Canada, in Guelph, in Ontario. Nobody knew what to do with it now. The humans are sequenced. What should we do with all this equipment? So therefore, now the barcoding initiative thought, hey, we can take now all these machines and use them just for simple barcoding. And therefore, the center of DNA barcoding is now somewhere in Canada, in Guelph. So all happy so far. Everybody thought, okay, we get now the huge diversity of organisms finally on the card. However, things are not as easy as they should. The problem is, if you want to barcode, if you want to understand what you have in your hands after you came back from the forest or from wherever else, you'd like to know what it is. You just want to use the DNA to understand what it is. You need the proper reference, which has already been sequenced, ideally, right? If you don't have that, then you have a new DNA strand, and you still don't know what it is. So far, there are still far too few reference sequences. So we have to build reference sequences which help us to have a backbone or a library against which now we can compare our now unknowns against. Otherwise, it won't make sense. So if taxonomy now is so difficult, understanding what is out there, how do we now get the best references? If I would now suddenly come with an earthworm or whatever else, and I would sequence it, and I'm not an expert on earthworms, I cannot say, hey, that's an earthworm. I put it into the reference because I'm not an expert. So what can we do to get the ideal reference points for the species for barcoding? What are ideal reference organisms for barcoding? If I have an earthworm, Lumbricus terrestris, or a classical fruit fly, Drosophila melanogaster, how do I know that that specimen I have is this species? Or how can I make sure that I feed my database with an organism of that particular species? Which specimen would you choose for building up a reference library? Any idea? I ask you guys. What? NCBI is full of mistakes. 10, 20% of NCBI is full of mistakes. People just sequence something, put it inside. There's no taxonomic check. I don't trust NCBI. We don't. Of course, there's nothing better yet. For sponges, we have something separate, where we still have some morphological checkup. But NCBI doesn't check. If you take a sequence which looks like an earthworm and you call it Lumbricus terrestris and you send it to NCBI, as long as it's within the annelids, they will accept it. And maybe it's something completely different. So what can we choose? What kind of specimen can we choose as a reference? Ideally, then we know 100%. This is the species. And I guess you all heard about that. Sponge. Why should a sponge help me to identify the earthworm? So, have you heard about holotypes? Have you? No? Not yet? Okay, good, let me talk about that. Holotypes are those individuals that researchers use to describe a new species. That single specimen, which had an unhappy day because it was collected by the researcher, was put into ethanol, and then subsequently described. That's the so-called holotype. Every species, every single from the 1.5 million species described, almost everyone, has a holotype. This holotype you'll find in species descriptions where when a new species description is made, usually very boring to read, but it says then somewhere type material, holotype, and it refers to this particular specimen. Where then researchers 100 years later can get back to and say, okay, Linné or Lamarck described that very specimen as a particular species. I have here another species that looks at a specimen. It looks the same. Is it the same morphologically or not? So the holotype is a reference point for every species. There's a holotype for earthworms, for Drosophila, Melanogaster, for the honeybees, for basically every species. Once described, there is a holotype. And this holotype, where do you find it? You find it in the Natural History Museum. This is Natural History Museum of London. You see, it is big and it's beautiful. If you're ever in London or in other cities which have huge natural history museum, Paris, Vienna, Berlin, in Madrid, or even outside Europe, I haven't visited that many outside Europe, these musea are cathedrals. They've been built as cathedrals because at that time it was thought that all the people, they have to be shown the beauty of what God created, thought 200 years ago about that. So therefore they are thought to be kind of cathedral to display what is out there. It's still beautiful to see. And beyond all the exhibitions, you see all these huge collections of animals collected from researchers about centuries. So this is a repository for biodiversity throughout time. So there are all kinds of stuffed birds is in these drawers, or you have this arrangement of beetles, and one of these specimens always has a red label. And this is the very specimen that Linné, Lamarck, or whoever else had in his or her hands to describe this particular species. That's a so-called type. And this type specimen is therefore Z.0 for this particular species. You find this usually with a red label here. For example, this particular sponge that you see over here, red label. We have a red lid here. Here, me subsampling somewhere in Amsterdam, some sponge. So they are therefore of incredible taxonomic value. And therefore, they are the most important organisms to build a barcoding library from, right? So, talking about holotypes. These holotypes not necessarily have to be complete specimens. It just needs to be something diagnostic that the researcher found and thought, oh, that's something different. This crumble here, you see here, yeah? The entire label here is, let's say, 5 by 4 centimeters. So you see this crumble is tiny, like that big here. There's hardly any material. But this crumble is already sufficiently diagnostic to say this is a complete new species, although just a small crumble has been found and not the entire specimen. This thing here is not an animal. This is just an ornamental piece, once found on a market in 1860, don't know where, in Central Africa. A researcher, Stanley, noticed this is fur coming from an animal not seen, at least to the Western, to the Western world before. And he collected it, he bought it. It is, you know, something like an ornamental, actually, I don't know what it is, what the meaning of that thing is. But he knew this must be from an animal we haven't seen before. And there were these rumors, there would be a kind of, how did they call it, a forest donkey, how people at the time called it, never seen before, but there must be something like this. And this tissue is coming from that. So he bought that and he directly knew this is a new species. You guys have any idea which species this thing is coming from? It's very characteristic once you've seen it. From Central Africa. Could be a zebra, but the stripes are not as regular as in the zebras. Pretty close. Taxonomically, not that close. It looks the same, but it's not. Huh? Liger, no, not even that. Not even that. No. It's the okapi. So this ornamental piece has been taken from the hind legs, but this thingy here is the holotype of the okapi. Okapi has been described by just this little piece of tissue from its hind legs. So it's one of the few animals described by its butt, actually. And the remainder, and much earlier, before the entire organism was ever seen. Still, this is the holotype of the entire okapi. This is a reference point if you'd like to extract DNA and build up a reference library. So, go to the museum, ideally, try to extract DNA from these materials. Okay, okapi is not so difficult, yeah, but other organisms which pretty much look alike, there's so much trouble identifying whether you have now the real species or not. So you have to go to the type specimen just to be very sure that you got the reference. Okay, so these guys were now collected 100, 200 years ago. Without that, we knew there would be something like molecular biology. So they've been dried, put into formalin or some things. And if I go out in a museum and try to collect prototypes of freshwater sponges, like in National History Museum of Central Africa in Belgium, you get something like this one here. That's a freshwater, it was a freshwater sponge decades ago, completely dry now, a layer of dust on it, quite some fungus on it, probably already rotten there for a while. Try to get DNA for barcoding out of that. Pretty much challenging. Or holotypes look like something like this one here, just some cross on a rock, yeah? Or they are associated with lots of labels where you have to understand actually what that means, yeah? And this tiny crumble in here is actually the holotype, yeah? This is a kind of a nightmare to work with. Or something is rather cryptic to get still DNA out of these kind of crumbles, and you have a huge rock, and somewhere there must be this tissue, and you don't know where actually this tissue should have been. So you see, it's not so easy, first of all, to get access to these samples. And then one big problem arises. You get the basics now, how to extract DNA, how to amplify things. Old DNA is different due to two reasons. First of all, what happens? The bases might be modified. How is that? You know the bases: adenine, cytosine, guanine, thymine. This is cytosine on here. Has an amino group over here. Yeah. Happens after a while. The amino group is lost due to addition of water, changed into ammoniac, and instead you get an oxygen over here. This makes out of the cytosine uracil. You know uracil? Yeah? Where do we have it? And RNA, all right. When you do the PCR, your polymerase doesn't care if DNA or RNA. It reads then this changed cytosine as uracil, and after PCR, it builds in the complementary, so the adenine, because you know uracil is actually the thymine in RNA. Therefore, the counterpart is adenine, and during then the synthesis of the opposite strand, hooray, we get an adenosine. So everything which was C before is changed into a U and subsequently will be read as an adenine. So it changes the genotype. So you have to be very careful to see these particular patterns. But that's not the only problem. What happens with DNA after time? So an organism dies. What happens? All proteins which are already in the cell try to lyse the entire cell. And what happens with DNA? Say again? No, it doesn't do the replication. No, definitely not. But what happens then with the entire strand? Will it stay like with your 6 billion base pairs per cell? Will it stay like that and twice 23 chromosomes? Or what will happen with it? What will happen? It degrades. How does it degrade? It will be chopped in pieces. It will be chopped in nasty small pieces. Here on the left-hand side, you know, electrophoresis, agarose, electrophoresis. Okay, cool. So left nuclear DNA extraction, which was not too bad, yeah? Lots of high molecular DNA, a little bit of smear. Well, we waited a little bit too long, doesn't matter. However, the further we get to the right, the more shorter the bands get, yeah? Here we have already almost entirely short DNA fragments, and if you have lots of bad luck, we hardly see DNA fragments anymore. Now imagine 200 years old specimens, of course. You do not get the long chains out of that anymore because it was all fragmented and chopped into pieces. Now, your barcoding fragment is 600 base pairs long, 650 base pairs approximately. If your DNA is chopped into pieces of less than 300 base pairs, or maybe 50, something like that, how do we want to get with PCR a long fragment of 650 base pairs? Barcoding will not work with that classical methods. Therefore, another possibility is to not take the entire 650 base pairs as barcode, but just take a smaller part. A reduced barcode just on something like 200, 300 base pairs, still sufficiently thick to say this is species A or B, if you cannot get any kind of longer fragment. This is also something successfully used so far. So far, mostly among insects, but also organisms. Next. This is our world. You see, is it? You see here differences, a plot of differences, so how much difference accumulate against the number of organisms. Ideally, if you sequence within one species, so given in red here, you should have a distinct range genetic distance. And if you compare against another species, given in yellow here, it should be also distinct. And in between, there's a gap without any kind of hits in between. So distinguishing, if we have genetic differences of a certain amount, we are still talking about the same species, we stay in red. If the distances is considerably higher, then we are in the yellow zone here, yeah, and therefore we talk about two different species. Unfortunately, the real world looks like this. There are some species which have a particularly low intergenetic distance compared to its intragenetic distance. And there we have overlaps in which then the variation within a species is actually already in the range of the variation between species. This is, of course, not fun for barcoding. We have that the particularly slow sponges or cnidarians, particularly corals, where then markers are particularly slow, as you can see here, compared to bivalves, so the mussels and arthropods and echinoderms. And you see then in this plot here in gray, intraspecific, so the variation inside, and the black interspecific, the variation between two species, we find here on the very left side always overlaps. That's the variation in some lineages is some species is as big as between species. So we cannot barcode that with this particular marker. Because the variability between the lineages is far too large, leading this to a problem depicted here. You see the barcode of 5 different species. These 5 species, however, you see they're all identical. And they're not only 5 species, they also constitute 3 different genera of different families. One big problem. This particular lineage for this particular group of organisms. Higher here. Yeah, please. Hello. Yeah, I guess better. So, yeah, because see when I move my head, suddenly things sound different. So with this particular marker, we cannot resolve our problems, unfortunately. And for some lineages, therefore, we have to take a different genetic marker for sponges or for corals. We take something else, but usually we know in advance if we deal with sponges or with earthworms. So therefore, the switch of a marker is not so much of a problem. So that means we have these methods with these problems for taxonomic barcoding. And there's a lot to barcoding than just taxonomy. Yeah, taxonomy certainly to understand, hey, how different are the individual species here. In the old days, taxonomic barcoding was done with restricted fragment with RFLPs. Now these days with barcoding, therefore we have this beautiful barcoding initiatives that help us. Just to mention briefly, these guys here, because we are running it, a sponge barcoding project, that has been used because sponges already 70 years ago, it was said that sponges are the last major order of organisms in which the sponges are the last phylum on which even on order level, we haven't had a clue what is out there and how they were related. Due to the fact that the entire taxonomy was only based on very simple skeletal elements, so-called spicules, which are arranged in a certain way, which can be sometimes a bit more informative, a bit more characteristic, but we easily run into homoplasics with the spicules. Yeah? You might think that spicule looking like this one comes from the same origin. Yeah, one rod here and then 2 discs here. Yeah, they kind of look the same. So, and you know, evolution is rather lazy. The chance is high that this has been made just one single time and then modified in a way, but no, unfortunately not. Homoplasy is pretty frequent. Or some of these elements just change wherever you put them in the sea. These spicules are made of silica. This is a spicule which is pretty much modified due to the fact that the species is living at bottom regions of the sea. There's higher silica. If there's higher silica, you get these strange modifications of all kinds of different spicule types. Here on the left-hand side, K is the way it looks normally. If you have the same species growing, however, in the depths of the ocean with more silica, it gets some very strange extra shapes. Like that one here, the picture below, A. At shallow depths, low silica, spicules, okay, that same species at greater depths, things look different. And for a morphologist, taxonomist, that's a nightmare. And then these things are arranged in a way with to form a skeleton. And you see this skeleton can be either echinating, or it can be, as we call it, confused, or it can be looking like these ones here in glass sponges, all right, a bit more complex. Yeah, different shapes here, very much rigid, interlocked, but sometimes the mineral skeleton can be absent at all. Then you have the bathing sponges, but you get the idea. 9,000 different species we cannot distinguish just with these few different characters. And therefore, we build up this barcoding project to help us to understand what's out there. We got a grant from MARBOL, Marine Barcoding of Life. Then we contacted our friends in the Queensland Museum to provide us with 11,000 different sponge species. And this lady, her job was to put 11,000 subsamples into small wells of 96 plates. It looked like something like this one here. And we went and extracted and barcoded and checked and did some sequencing. And out of these barcodes, finally, if you have lots of data, well-established data, you can answer many, many phylogenetic questions. Once you have lots of data, you can put all this data in a huge tree. Then you can start answering your questions, such as, for example, like that one here. We can, with this data, reveal endemism, finding that every single bay, now, for example, in Australia, has a kind of endemic fauna, species which are just there, and the same looking species in the next bay might be something different, cryptic speciation, so-called. Genetically different, probably a different species, and this basically everywhere where you look. See, the level of endemism, for example, is higher, and you find this easily with barcoding approaches. Then cryptic speciation in the deep sea. It was for a long time thought that in the deep sea, genetic distances would be large. No, they are not. In the deep sea, on the bottom of the sea, you find that there's much more interchange between species over long distances than at the surface. And you only reveal that if you have the genetic data, when you suddenly notice that there's not only cryptic speciation, so much more going on there in terms of speciation, but also that some lineages are actually identical independently if they are somewhere at the southern Mediterranean or in these Atlantic trenches, or maybe over here. Likewise, ideas about water sponges. For example, how colonization of so-called ancient lakes originated. Ancient lakes that are hundreds of thousands of years old, with obviously an endemic fauna. Where is this endemic fauna coming from? For a long time, it was thought that it somehow originated on its own, but no, that was not the case. Instead, it is that, given here in yellow, that an endemic family that was thought to be just monophyletic is not monophyletic at all. Instead, from there, all kinds of other lineages split off to colonize the ancient lakes and become with this endemic. This was pretty new to science at that time. Or more applied. Last Wednesday, we talked about all the biochemical compounds, right? That sponges produce. Now, we could show that some compounds, which were believed to be separately produced in distinct crates, are actually only once produced in one big crate. And our taxonomy, the so-called families, how we called them at that time, were actually not valid at all. And other members of this family that do not produce these compounds, group somewhere else. So with the molecular data, with the barcoding data, we now could find how frequently these particular compounds originate, how frequently all the genetic modifications necessary to produce these compounds are established. Likewise, another example here, this one over here, this molecule over here. This is a certain order. These are organisms from a different order. For a long time thought that they might not be related to each other at all. But this compound was produced independently, of course, from all lineages. But no, they form a beautiful monophyletic group. And all other organisms that should have belonged morphological to this group do actually not join this group and produce a different compound. So with this, with barcoding data, ideally, you can answer lots of other questions, not only phylogenetic, but also methodological. And this is then the big power, of course, of these barcoding approaches. Likewise, you can do other fancy things, like environmental sequencing. Yeah, you might also have heard about eDNA, where we get actually the chance, we just sample a drop of water or whatever else is in the environment, and we clone it, or we do other measurements with it. And with this, we directly find what is inside this particular drop of water in terms of organisms. And a nice example for that is to understand food chains. Yeah, big fish eat smaller fish that eat even smaller fish that eat krill. But what is eating the krill? So identifying what krill eats is, of course, rather nasty and rather difficult. So why not just open up the gut of the krill and identify directly what its diet is made of? Yeah, the gut contents, just check. You see here all these OTUs, operation taxonomic units. We can then associate directly what are the food particles, or what is now the overall diet of the krill. We don't need the full organism for that anymore. What else? You remember that 10 years ago, the spectacular emergency landing of this American or US Airways flight in the Hudson River. There was even a movie of that, Sully called, yeah, with Tom Hanks. Fortunately, everyone was saved. And the cause of this was certainly a bird strike. Okay, so far, so spectacular. However, it would be still cool to find what the unfortunate organism caused the bird strike. All airports have biologists that should warn the pilots, hey, there's currently a flock of birds going that direction, or this is now the major migration time of that, so we have to take some measurement. So we need to know what organism went then into this particular engine. Of course, you can imagine there's not so much left of this unfortunate bird, but these remnants, of course, are more than sufficient to compare, to extract DNA, amplify, sequence, compare against a well-established barcoding reference, and to understand which of these poor geese was actually causing this trouble, and hopefully prevent that this will happen in the future. Once in Australia, I found this label in a bus. Spitting is unacceptable on transfer services. So obviously, for any reason, they have some problem with people that spit on the floors. And obviously, they have already DNA kits to do all that magic, to identify who was spitting. This is also taxonomic barcoding, yeah, just to find, however, who did all of the mess. I was pretty amazed to see something like that. It's pretty, quite a high-tech country, no doubt. So, except that, yeah, certainly we find with taxonomic barcoding, food webs, larval stages, dimorphism, all these things you always wanted to know about an organism, but never dared somehow to ask. We can get a bit more applied to that. For example, we can use applied barcoding to identify commercial fish and to detect food fraud. So there's been, in the last couple of decades, this is already a pretty old chart, but it's still actual. You see that throughout time, the fishing, the overall catch increased steadily, continuously, until in the 90s, there was a kind of stagnation. This stagnation certainly leads to quite some trouble. This stagnation is, as you see over here, although the amount of people on Earth has been rising and the amount of fishermen has been rising that time, the overall catch per fisherman decreased. That's not good for the fisher because they need their money, of course, and prices are rising, of course, for everything. So what happened? So the fisher went out and looked for new regions to catch. Here, as an example for, I guess, for a certain codfish, if I recall it correctly, you see here the word graph. The warmer the color, the more has been caught per 100 hooks right out there. Everything which is blue, hardly anything collected. Everything which has warmer colors, this is so far still pretty much all right. This was 1952. You see already these regions around Japan, for example, where there is not so much caught. But if you go throughout time, we see that the fisher and the regions where fishing has been conducted went more and more to other regions, while the original regions where has been fished, there has been hardly any kind of productivity anymore. The colors get cold here. New regions have been sought. Now getting into the Atlantic, where still has been lots of fish, but the further we get, 1962, 64, you see the colors are getting colder, and basically all regions on the world are now kind of, at least temperate tropical regions, therefore, are now assessed. And you see at a certain moment, everything has cold colors here. So hardly any gain of fishing here anymore. Clear sign for overfishing. Oh, it goes on. There we go. As an example, Atlantic cod. Yeah, there was suddenly a huge demand, which led to incredible overfishing. And suddenly there hasn't been anything caught anymore in the mid-80s, early 90s. Then there's been a race again and a certain drop again. So that means there hasn't been any success for the fisher to collect fish anymore. So still the fish are around, mouths to feed, demand on the market was still there. So what do you do usually? Either you sell less or you, let's say, you modify your results, your fishing results. There's now high demand of these organisms anyways to eat. But of course, there's also increase of public awareness that fishes are overfished. So the solution was to give the impression that this overfished species is still in common and do that with false fish declarations and maybe even mislabeling. So mislabeling of fish. Usually identifying fish is rather easy. Yeah. If I see a chart like that, I could identify fish, even I would know something about fishes. Identifying fishes here is a bit more tricky. Yeah, I cannot do that. And if you have these already as ikan goreng, yeah, or somehow else fried, yeah, try to identify that fish optically, so it doesn't work. So what you do then is you take genetic methods, barcoding. And the Fish Barcoding of Life initiative helped a lot with it. And based on this, there were some amazing results, or shocking results, actually. This is a pretty popular fish, the red snapper. And there was a study that checked what happened with the red snapper over time. And all those animals which have been declared as red snapper on the restaurant menu. Are they red snapper or not? And here the phylogenetic tree on the right-hand side. Red snapper in red, conveniently, and all the blue are those samples collected from restaurants or from the markets. And you see only these samples here, samples 13, 19, 17, 15, 4, I would refer to as snapper, and all others are red snapper, and all others are not red snapper. So this is fraud, actually. And even stranger, sometimes the mislabeling went so far that they even have taken another completely overfished species and took that one instead. So in another study here from 2008, on markets, 99 labeled fish as snapper were not red snapper, but even 56 of these not red snapper were already overfished species. This is not fun. Then, of course, you just move the problem somewhere else. So we took this as an example in Munich. We come to this example later. Other examples for fish taxonomic barcoding is that we had in Japan some cases that people, after eating monkfish soup, got in serious trouble, where then it's been noted after DNA analysis that the monkfish was not monkfish, but instead it came from the pufferfish, which you know has one of the strongest toxins of vertebrates and maybe of all marine animals at all, namely the tetrodotoxin. Simple barcoding already indicated that these samples are big in this black crate here, which is consisting of all these pufferfishes, while the original monkfish is somewhere else, shiny happy. So, well, that means I hope that the patient survived. I don't know about that, but anyway, barcoding helps. And therefore, we tried the same for fun, for teaching studies, for teaching students in Munich and going out on the most popular market in Munich, the Victualian market. It's a traditional market there. Ask them to buy some fish, some parts of fish fillets, etc. And we went out and barcoded it in courses. And by the way, this gentleman you see here is 10 years ago, Dr. Edwin, who was helping in this course as a teaching assistant. Yeah, so we therefore took these samples and did all those things you've been taught now by Dr. Maya, getting the samples, extract DNA, amplify, clean up, sequencing, whatever else, yeah, and then try to see what it is. We did not find food fraud, but we found some pretty interesting labeling facts of fishes, people that were not aware of, but at least it's a pretty nice way to understand how barcoding works and how useful that is. Yeah.
We also used in the halals, in the framework they use, but not for the fish, but Professor using the barcode one. So you can imagine now what is from the standard records of halal. Is it fish? What? Now, what is the standard reference of the barcoding in the halal? Pork.
So you check for traces of pork in food?
Yes.
Yeah, okay. That of course can.
Halal sample is also the prominent center at ITS, so we should understand.
Oh, okay.
Yeah.
So
okay. They have already checked, yeah. The PCR.
Not only for the PCR, but also from the chemical compound used by.
Okay. Meat. Okay.
Check what is inside the meat.
Good.
Okay.
Food fraud. Good. Of course. Go ahead.
16 as regime will be divided into 4 or 5 regions, and which of these regions can be chosen as the barcoding item?
Well, yeah, there's no unification yet, which region to choose when they are shorter. Usually you take the, if I recall correctly, the V4 region of the 16S for barcoding. Yeah. One of those, yeah. How long is that one, the V4 region? Yeah, okay. But for bacteria, you don't need the shorter barcodes. Yeah, because for bacteria, you can make cultures, you can grow them, you have directly sufficient fresh material to work with. So the need to get back to very short barcodes is not given so much in bacteria. Yeah, so in this respect, I don't think that this is a question that is currently prominent for bacteria. Yeah, these very short barcodes are just if you have tissue out of which you cannot gain any kind of additional DNA anymore. Yeah, and this is not the fact for bacteria currently. So I wouldn't worry too much about that. Okay, good. So now we can also identify introgression. Phonetic introgression, for example, of the marine systems. Yeah, the world traffic rises, continues. There's cargo ships going everywhere. And in the marine realm, we even find that very frequently new organisms are introduced and replace the original fauna, causing quite some phonetic trouble there. So how can it happen? Either by the ballast water of the fish, of their ships, yeah? In one port, they pick up lots of water to be sufficiently stable, and then they return to the other port. They release the water, and they can pick up the freight, so they're still sufficiently stable. So the water of Ocean A is getting into the water of Harbor B in Ocean B, and therefore leads to the fact that new larvae and new organisms get there. Likewise, hull falling. So the lower side of the boat, which is full of algae and bivalves and other things which are growing there. So with this, lots of other organisms can be moved from one port to another, leading therefore that very aggressive specimens can make it from A to B. The trouble is that most of them, these introduced organisms, are cryptic species. They look the same. They occupy almost the same ecological niches, but they're different in their lifestyle, in their need of food, and in their reproduction, probably much more dominant, and maybe they're much more harmful. But we cannot tell them apart. From all these yet identified, from all those recognized introduced organisms, most of them are yet undetermined. So you see in black here, these are the undetermined introduced organisms. And you see there's actually quite a lot there in terms of cryptic species which are introduced. Nice example here are ascidians of the genus Clavellina. Here, a nice part of the Mediterranean Sea, somewhere where these beautiful ascidians are growing. And genetic data, however, showed that a species, a much more aggressive species, which is coming from the Atlantic side, is now invading in regions of the Mediterranean ports. Inside the Mediterranean not port regions, you find the organisms of this genotype here given with a green box here. The Atlantic ones, which are much more aggressive, you find over here, and the same genotype you find around Mediterranean harbors. That means with high likelihood, all these organisms are brought en route from the Atlantic to the individual harbors. And from there, they are slowly colonizing the harbors, and from there they're spreading out and cover the remainder of the shore. And they're getting so very aggressive that they're not stuck to just the Atlantic. Some publications now show that they make it all the way up from Canada to South Africa. And this is therefore probably getting a kind of colonization into foreign biota, into a new biota, and therefore replacing the original biota. And therefore, these guys have already been suggested as a good candidate for undertaking immediate eradication to prevent further spread. You can imagine, logically, counting these organisms apart is pretty difficult. But barcoding helps to directly identify this is a new introduced species, and we definitely have to do something against that. As a last part here, evolutionary barcoding. And evolutionary barcoding is you take your sequences, you make your data set, you use a bunch of fancy algorithms you're still going to learn or you know already about, you make hypotheses, and out of them, you answer a bunch of questions. And see, some of these questions might be of immediate need due to the fact that there are some threats for our coral reefs. You know this organism? Yeah, the chronothorn starfish. What does it do? What does it do? It's pretty nasty.
All goes wrong.
What does it do? It suddenly appears in coral reefs. You see it over here, inside a healthy coral reef. It is unlike all starfishes. It looks slightly different. It has spines with toxins, with pretty potent toxins. Unlike other echinoderms, it can have 14 to 21 arms, so with a pretty huge surface and up to 1.5 meter in diameter. Running on tube feet, as you see it over here, and can be pretty fast. So with this, it walks over the coral reefs. And what it does is certainly it tries to eat off everything living of the coral tissue. So how does it do that? First, it senses here's a coral, then it extends its stomach over the living coral and digests extra-intestinally all the tissue, and it sucks everything up again and can do it in an incredible fast speed due to its size and also due to its speed that it can move. It's actually pretty fast for a starfish. It can, within an hour, it can make 20 meters. So that's actually quite a lot. So within shortest time, it's therefore able to change an environment of coral reefs, as you've just seen here, from nicely living to suck up everything which is alive and leave therefore everything just naked coral skeleton. So therefore, these things are identified as a major threat because they're not only fast, they're also very fertile and very potent in reproduction. You know, that means they produce lots of eggs at the same time, yeah, out of which then lots of new starfish originate. So they can therefore chew up a coral reef within shortest time, and these outbreaks are therefore strongly monitored, and fishermen have to go, volunteers have to go out and collect them all one by one to save the reefs. The only thing that really would prevent these kind of outbreaks is, of course, the natural predators, like the triton snail here, which eats on the chronothorn starfish, or other fishes, such as here another snail here, like a parrotfish, or a pufferfish, or triggerfish, if they would still be active in the reefs. But also they suffer very much from overfishing. So that means that natural enemies, the natural predators of these chronothorn starfish are therefore decreased, are therefore going to be extinct, and therefore the chronothorn starfish can therefore spread out in outbreaks, in regular outbreaks, and therefore chew off all the coral reefs, with huge, of course, economic problems. It looks like something like this one over here. Here you have here a pretty nice, still living coral, and here everything is chewn up, munched off, and here you still see the crown of thorn leaving the crime scene after eating off everything here, and now trying to get to the next coral to feed on. However, they look kind of different, as you see here on the left-hand side. Some beautiful in blue, some not so beautiful in brown and greenish, whatever. So it must be an imperative to understand what is the species structure of them, to understand, are there now one big species, or are there lots of different species with their individual timings of outbreak, with their individual parameter when they go out in the reefs and cause all the trouble. And this is what we did a couple of years ago, where then we barcoded these cornerstones, noticing there is actually not one single species as suggested here before. Instead, we find here 4 distinct lineages of the Pacific, Red Sea, Southern Indian Ocean, Northern Indian Ocean, nicely separated from each other, mostly nicely separated from each other. And each of them is very distinct. And each of them, therefore, might have their individual moments of outbreak, of reproduction, and other parameters. And here we can start and say, hey, this is species A. It does at this moment whatever. We have to take care of that at that very moment. Species B and C do not do anything yet, so we can wait, so we can focus on that particular species and make sure that the coral reefs stay safe until the next outbreak. So based on this data, we could therefore make some measurements crucial for the design of all kinds of other management strategies to reduce the chance that we have catastrophic outbreaks of this crown of thorns starfish again. Meanwhile, there was a chance, and therefore, that some of these species got already now their own names, with their own holotypes, with their own references, with their own reference data points and data sets from which we can control then all kinds of further outbreaks here. And with this, I guess, and I hope that I provided you with a little bit of overview on what we can do with those techniques that you are learning here, that you applied here successfully, that you hopefully understood completely what every single enzyme does at which particular moment. Very important to apply that at all. And you see that DNA taxonomy or barcoding is not just knowing, hey, this is species A, this is species B, but who cares about that? But this has quite some important consequences and some important applications for our living system. All right, so much about me. Questions? Thank you.
Yeah, is it any question? This is really interesting. 1, 2, 3, 4, 5. 1, 2, 3, 4, 5.
I better get a seat.
6. How is the rule, ya Bu Maya? Once answer or?
Just go ahead.
Yeah, maybe once first from the first one. Yeah, lady. Yeah.
Check. Okay. So thank you for the chance. My name is Lady. Actually, this is my first time ever hearing about the holotypes. So I want to ask about how can we practically determine that the particular parts of the animal can be used as a holotypes? Oh.
Again. The question again. I didn't get the first part of the question.
Oh, no. I'm just saying that this is my first time ever hearing about the holotypes. How can you determine practically an animal to be a holotypes, or the particular parts of the animal can be holotypes?
Yeah, well, of course. Thanks for your question. Very good question. So you are a taxonomist. You know everything about your beetles you so far worked on. Then you go into the forest or wherever else, and you suddenly notice there is a new beetle, something you have. You, of course, take this beetle, you compare it against everything that you've seen, which every other beetle species in the literature, and you see, now this has not been recorded before. Then you take this particular organism and you use this for the description of the new species. Now it can happen. You do not see one single species specimen, but you suddenly have lots of specimens. Yeah, of course, you never see one beetle, you see a lot. There's a population of beetles you haven't seen before. So you therefore choose out of this one, which one is the best to pick? All look the same, you just grab one. This will be your holotype. Hopefully, it has all the legs, ideally. It should have all the legs, all antenna, or whatever else. It should be as complete as possible. But it does not have to be complete. Sometimes it just needs these few features that distinguish this beetle from anything else, like the okapi. You just have a piece of tissue from the hind legs. This is a symbol. We are different from zebras, from giraffes, from whatever else. Nothing has this kind of pattern. Of course, then to describe what the okapi looks like, you need an additional specimen. Yeah, this is then brought to you, or this is then mostly then described a little bit later. But the holotype itself should be something where, say, this is here diagnostic, and I describe now my species based on this very specimen. And therefore, you choose yourself which ones, because you, in this moment, the very expert of this news that sees it. Yeah, you pick this species and say, this individuum will be now my holotype. By the way, all other individuals that you see in that very moment become the so-called paratypes. Also very important. These are not those that are directly described, but they are collected together with that specimen, which is becoming the holotype. So if the holotype is lost, or you cannot find it, you can take a paratype instead, because it's probably the same species, because it was found in the same moment in the same area, and therefore you can use this for all other further analysis as well. But first, you find something new, you compare it against whatever else is known already, and if you know this is new, then you can say, use the holotype and you describe. Does this answer your question?
Yes, actually I can help you also. Actually, for type specimen, it will be taught for the master student for biosystematic. So actually for systematic, for bachelor level, maybe Bu Dewi or Bu Dina has not explained you about the type specimen like Professor Erpenbeck say. Type specimen consists a lot of things. So only not single specimen, but you need comparable specimen. So it like Professor Erpenbeck say, it should be series. So you will learn it later on the biosystematic on master, but in fact now you have to remember about type specimens, and only one, the most important thing is holotype. Rather I will explain about paratypes, what is, but it takes time. That's it. So for us in the bachelor level, it's only alpha and beta taxonomy, not until full systematic like gamma taxonomy. So that's the chronology. So yeah, okay. I hope you understand a little bit. Yeah, lady, at least keep on. You can Google it, type specimens and holotype. That's explained by Professor Erpenbeck today. Okay, thank you. The other Lucas, yeah.
Thank you for the opportunity. My name is Lucas Egidius. I'm third year student in the IDS. And here I have one question that, as we know from the presentation about the holotype problem, whether ancient DNA can be degrading or fully modified by this time. And is it still possible to describe the species origin, or is there any possible way to describe the origin with the other method? Thank you.
We see, Ali?
With the other method, I mean, with
Yeah, is it only because this is only small fragment, and is it any other method to describe without molecular DNA, you think? Yeah, so you have only small fragment, then is it any other method besides using barcoding, because it's only a small fragment? By morphology or something like that, you mean?
You mean during ancient DNA, when you have
Yeah.
really old DNA
Yeah.
and you do not get that fragment
Yeah.
that you'd like to have? Yes, there are methods which put together only those very short fragments, out of which you will be able to get, actually, when you are lucky and you have lots of these short fragments, maybe even entire genomes. There are methods in next generation sequencing which give you the possibility to just put together, by comparing against a given reference, putting together all these small DNA pieces and sequence and getting out of these small fragments then actually the full length of this particular region you're interested in. I was thinking of introducing that here, but I thought it might have been a little bit too early to introduce it here. But to make a very long story short, yes, we have the chance to get out of very degraded DNA, consisting out of just 20, 30, 40 base pair long fragments, still everything we'd like to have, or not everything we'd like to have, but many data that we'd like to have for barcoding or for other fragments. Yeah, so it works nicely.
Okay. Thank you.
Molecular method, there is also another. We only have a small part of the species that we want to identify.
Morphology. Yeah, but other than DNA, I wouldn't know. I wouldn't know. Oh, yes, I do know. The time machine.
Next. Yeah, time is really.
Okay, thank you, sir, for the attention. My name is Satrio, and I have several questions in here. And the first one is, in your presentation, you just explain about basis mass may be modified, DNA may be degraded, which is we know the species have gone or died, but how we know that the DNA still exists, or how we explain it, how we know it that the molecular still exists, and I mean, how do we know about this species or this one is, we know about it, but they have died, and maybe the basis or DNA have gone something, but they are maybe still particular are bit. So how we know about it when this organism or one species is died, but they still DNA. And the second one is we know about the species, many species, one species, or I mean maybe Homo sapiens. Why they are in the still genus or species, the same genus or species, when they have different morphology or have different molecular basis. I mean that. Thank you for the attention.
All right, thank you for your question. Actually, a very good question. The first one, whether we know that they still have DNA or not to be used, we just have to try. We don't know. We don't see in a corpse whether there's still usable DNA or not. You just try to take a little bit of tissue. Don't take too much because there's otherwise not much left of the organism. You don't know in advance whether this will work. Then you still don't know how much of the bases might be modified. Yeah, you were talking about the change from Cs to Us. You know that from the very beginning. There are, however, a bunch of algorithms that give you, looking at the DNA sequence, the likelihood how frequently that could have happened, and provide you with a bunch of suggestions what the original genotype might have been. Okay, before we don't have the time machine, we will not know if that really was at this particular places, but with these models, we try to approach that as much as possible. The second question is pretty good. So, how do we know that it's a different species or it's a different genus? We don't know. What's the definition of a species? Was it already a topic, Edwin? Yeah, species concept.
Species concept given for the master student.
For the master students, not here. Okay, okay. There are, to make a terribly long story short, there are something like 60, 70 definitions of a species. Morphologically, genetically, evolutionary, reproductively, whatever. Yeah. So you can define a species based on your morphology or just by the fact that they are sister groups, molecularly, whatever. So there is actually no consensus yet what is a species. We would now say mostly a species are those organisms which are reproductively separated from each other. This is something that we can find with the molecular data that we see. There is no genetic introgression from one group to another. Then you can say these two thingies are two different species. What is the definition of a genus? Okay, you haven't had species concepts, then probably you don't have. There is no definition of a genus. Every taxonomist, so when you are going out in the field and with the beetles, yeah, you can say, hey, this looks different. I call it a different genus. It's up to you. Yeah, there is no distinction when, not even with molecular data, when we are leaving the species level and we're reaching genus, family, order, whatever. This is still not somehow organized. This is up to the discretion of the researcher. Yeah, this only thing you can do is try with molecular, maybe also morphological data to see these things are sufficiently different that I assume that they are genetically not intermixing with each other, yeah, and therefore there must be, due to reproduction, single species. You can do this molecularly rather more straightforward than with morphology, but if this is constitutes just a new species or maybe even a new genus, it's up to you. Okay?
Yes, because Satrio, maybe you just remember on the bachelor level, you just explained by characters. Do you remember on the animal systematic or plant systematic, the character? So it's only based on morphology species concept. Maybe it has not explained further. And also, besides that, a species is an organism who can breed each other and then produce a new fertile descendants or something like that. So it has a lot of disputation on species, but you will learn deeply about that on the master level. But in fact, you know, it's only the characters. So that's what they learn on the bachelor level. Okay. More question? It was 6 or 5. Maybe
don't be shy.
Don't be shy. Oh, cool.
Thank you. My name is Adiputra Tidutan Rizky. I'm from ITS. Yes.
This comes as a surprise.
I want to ask you about the methods about and tips about the identification about the holotypes, and are these holotypes used only for complex organism, or how about the bacteria, virus, and algae? And I have a one worst case about these holotypes. And when we are in laboratory, something there are contaminant about the humans maybe, and we can identify the bacteria contaminants in the isolate with the holotypes or not. Maybe like this. Thank you.
Good question. Actually, the contaminants on the holotype, or and also, yeah, you can say about the contaminant and what the holotype for the small organism or microorganism.
Okay. So the holotype system was, of course, first only established for multicellular organisms. But of course, it does make sense. Yeah, there should be reference points for all kinds of organisms. So therefore, they are established culture institutes with culture collections. And these culture collections, they have strains which are references. And they breed these strains, they regain these strains, they keep these strains alive as much as possible. And these ones can be then designated holotypes. Of course, Escherichia coli, yeah, as a standard bacterium, has been known before the holotypes system was in place for bacteria. So what you can do then subsequently, if there is no holotype established yet, you can assign subsequently another organism or a culture of organisms. Yeah, mostly bacteria are clonal. That means if you have one, you have identical genotypes for the entire colony, and designate then this particular colony, yeah, as the new holotype, the so-called neotype. Yeah, there's then probably a bunch of people get together and they say, okay, for Escherichia coli or whatever these bacteria, we take this lineage somewhere, deposit it somewhere as the holotype, as the so-called reference point here. Yeah, so you can even subsequently assign holotypes or type material to individuals or to colonies if they haven't been assigned before. Yeah, so that works actually pretty nice. So you can therefore check references for strains, not only bacteria, also protozoans or fungi, for which you can get actually references. They are then in institutes all over the world. Good. Then your second question was how to identify human contaminations. It's very straightforward. You sequence something, and suddenly your DNA sequence is closer to Homo sapiens than to all other organisms you would expect there. You don't need a holotype for that. If you are sequencing earthworms or beetles, and suddenly there's Homo sapiens, yeah, so the genetic difference between Homo sapiens and the beetles is so large that you do not have to compare against a reference database. You can even take the bad entries from NCBI, where then it's clear that it was rather you who contaminated, or your colleague, of course, who contaminated that. Yeah, then this isn't the true bacteria or beetle or whatever DNA. Yeah, so don't worry too much about that. And another thing, talking about Homo sapiens, of course, there is the trouble with who is the holotype of Homo sapiens. It's a very complicated story. Yeah, so what should be the holotype, the reference point for Homo sapiens? Now we're getting into trouble. Yeah, Homo sapiens, of course, is it a man? Is it a woman? The holotype? Is it Asian, African, European, blonde, blue eyes, whatever? So it's a very sensitive story. So therefore, we so far refrained from taking a holotype. When in 2001, as the first DNA sequences of Homo sapiens were published, they were made of 6-7 individuals that donated blood and tissue. And actually, they should have remained anonymous. But one of these guys, Craig Venter, he is actually one of those persons that pushed forward all the sequencing initiatives, said, hey, I'm one of them, and this is now my genome. Now you can see that I probably will get, when I'm old, problems with cholesterol, and I'll probably get Alzheimer because you see it in my genome. Why not taking me? So there was a suggestion, we could take him, for example, as a holotype for Homo sapiens. Now we have the genome. Of course, it's a big no-no. Yeah? A white man taking as a holotype for Homo sapiens in these times, this is not a good idea. So we keep the discussion of who is a holotype of Homo sapiens a little bit underground. However, there is one type specimen for Homo sapiens. And this type is not a holotype, it's not a paratype. We'll learn later about that. It's a so-called lectotype. A lectotype is a specimen which has been taken by somebody to understand what the organism looks like. The Okapi tissue was a holotype. The first really seen Okapi was the lectotype. So you can describe, oh yes, it has 4 legs and 2 eyes and whatever it is. So we can go back and see what specimen has been used to describe Homo sapiens. Who described Homo sapiens? Linné. Who was around in the time of Linné? Probably his wife, probably his parents. However, we don't know for sure. We, however, do know for sure that Linné knew about himself. So he must have taken also himself to describe Homo sapiens. Not as holotype, but to describe also feature of Homo sapiens. So in this respect, Linné, buried in Uppsala in Sweden, is a type specimen of Homo sapiens, but clearly not the holotype. Okay.
The question on the YouTube. Oh, I once read that there is a research regarding DNA barcoding of macroalgae, which can be a bioindicator of heavy metal pollution. DNA barcoding work to find a suitable bioindicator. Is it?
Barcoding will not work as
this is from
bioindicator. So first, thanks for the question. The question was, I once read that there is research regarding barcoding of macroalgae, which can be a bioindicator of heavy metal pollution. How can DNA barcoding work to find a suitable bioindicator? The DNA barcoding will not work for finding a bioindicator. However, if you see that an organism is a bioindicator, barcoding will help you to identify what it is. And then you're not taking the classical cytochrome oxidase 1. Instead, you take what is used for algae, some chloroplast genes, RBL, RBC, something like that. And this identifies you what that is. But finding a suitable indicator, so all the chemistry and to see what is accumulating now all kind of heavy metals or whatever else, it's nothing that DNA barcoding alone can do. And I hope this answered your question.
Okay, thank you. Any more or no?
It's okay. It has been already published dealing with the database of your barcode sequence. For example, NCBI, we can take many sequencings from the NCBI. How about you working with your project and you already create the barcode, have already published one, we can download it easily, the barcode from your website? Huh?
Of course. Definitely.
Okay, what's the name then?
Spongebarcoding.org.
Okay,
yeah, so,
Sponge barcoding.
Which is currently a bit refurbished to make it a bit more user-friendly. But anyway, of course, every single sequence which is ever generated should go to NCBI, no doubt. Yeah, because this is a big repository to get all kinds of sequences from. So everything we publish for sponge barcoding, we send to NCBI as well. So because it should be out there. Yeah. However, this doesn't mean that every barcoding sequence somewhere produced in NCBI is a good reference. Only those that come from us, of course. Actually, they don't. Actually, they don't. And the barcoding of life initiatives, unfortunately, they grab lots of data directly from NCBI. They say they grab everything which is, for example, from sponges and say, hey, this is here the barcode, although it's never verified. So this is a little bit. So therefore, we encourage, if you want to be sure, yeah, take them from our side, download them directly there because they are curated. While on NCBI, this curation doesn't take place. Of course, we mirror our data is going to NCBI just to bring it to the public domain with this as well, of course. Yeah, we want to play with rules. But of course, we only keep those which are nicely curated with us, and you can download it from there.
Thank you. I think it's enough. I think almost 11. No more, yeah? Any? So if something, yeah, you curious, you can email me or email Professor Dirk, but directly you just tell ITS student of Edwin or something. So I hope it will be it will help to open your new horizon or new perspective, how important the barcoding. As far as I learned before, yeah, it is only for distinguish the space, but you know it. There is some other benefits also, like for the practical thing. At ITS we have halal detection, and even if you read in the newspaper, Indonesia has not yet good facility for halal, but non-Muslim country like Thailand or the other, they have more sophisticated barcoding method to detect the remnants of the pork or something like that. You can Google it. So it's a good opportunity for you for learning what is the benefit. Yeah, bisa. I think it's enough. Yeah, thank you. We give applause to Professor Atuan. It's ready.
Pause. Of course. Okay. Let us switch it off, okay?
Yeah, we just like it.